As you'll see from the rest of this talk, I'm very excited about the ISBM meeting this September, and I have been for many, many years. So with that, today's talk, I was asked to talk about sort of the background of shifting direction of the research program rather than talk directly about the research. So it's going to be a little bit of research in here, but really it's about, how do you shift gears and integrate something different into your research portfolio. And again, thank you. I see some of you are logged in from Europe and we strange hours. Thank you so much for joining us. Okay, so first thing I want to do is remind us that we're focused on musculoskeletal diseases, and these are some numbers from the US. 3 of the 4 most common chronic diseases in adults in the United States are musculoskeletal diseases, osteoporosis, osteoarthritis, and low back pain. And the direct and indirect costs of dealing with these diseases in the US are estimated to be $980 billion per year. But if you're not American, you may not have some concept of what that $980 billion means. Well, that's 5.7% of the gross domestic product of the United States. And if that's kind of sort of a vague number for you, the entire US Department of Defense, US military is 3.7% of the gross domestic product. So one of the largest militaries in the world, the country is actually spending more money dealing with musculoskeletal disease than maintaining that large military. Another couple facts I want to point out: those of us who study bone, we often people think about bone. Oh, it's whatever this disease is, not deadly, or it's not as deadly as some other diseases. This is the true mortality that you get, the 1-year mortality. So if someone experiences a hip fracture, their 1-year mortality is 30% odds of dying in the year after the hip fracture. That's approximately the same percentage risk of dying if you're diagnosed with stage 4 metastatic breast cancer. So keep that in perspective. Another issue that I hear more and more about is antibiotic resistance as a threat to human health. And let's put, again, those numbers in perspective. In the United States, we have 0.3 million hip fractures annually, resulting in 31,000 deaths. The number of deaths from antibiotic-resistant infections, or number of antibiotic-resistant infections, substantially more, but approximately the same number of deaths per year in the US. Okay, so why are we here? I went through graduate school, extremely interested in bone remodeling. This was a figure that I spent a lot of time thinking about while I was a graduate student, the interpretation of the basic multicellular unit in cancellous bone. And it fascinated me how we could, from 2-dimensional sections using dynamic histomorphometry, figure out this entire structure. But I wasn't satisfied with that. And when I started my own laboratory, we kicked off in 2005, we kicked off 2 main research goals. And one was to actually visualize these basic multicellular units in cancellous bone. And so we developed our own image acquisition and visualization approach. These are some of the images. We presented them first to the ISBM in 2009. But what you're seeing, the colors are bone formation markers in rat bone, calcine and xylanol orange, spaced a certain amount of time apart. And you can see the resolution of the surface of the bone is such as you can see the resorption cavity inside the bone. And so we were able to make some measurements of the BMUs. And we're very proud of our ability to take these segments of trabecular bone and look up close at locations of bone formation and make measurements that weren't possible with the 2-dimensional dynamic histomorphometry. We also expanded on this and looking for other ways of applying it. This is a study we did where we used an in vivo mechanical loading model to load the trabecular bone inside the rat's tail. And then we combined our 3-dimensional imaging of bone formation, which you see here on the left. That's the green is the bone formed after the mechanical stimulus to the animal. These are the locations of osteocyte lacunae, which we got using a higher resolution microcomputed tomography. And then on the right is a mechanical model, a finite element model that shows us the distribution of strain energy density and mechanical stress. And we were able to make some correlations among those things. And lastly, we're very proud of using the same imaging approach to look at microscopic damage in trabecular bone. And this is one of our studies. In this case, the colors, the orange and green are regions of microscopic damage in the trabecular bone that we cause. So we were able to study how the cracks, or approximately the cracks, damage regions in the cancellous bone grow over time as the trabecular bone submitted to cyclic loading. And so we're very proud of this work, but now my laboratory looks very different. Now my laboratory, about half of the laboratory focuses on the effects of the gut microbiome in musculoskeletal conditions, including bone. We do most of this work in mice, looking at bone mechanical properties and geometry. We also do work looking at osteoarthritis, and we've also done some studies looking at susceptibility to infection in animals after an orthopedic surgery. The other half of my lab is focused on microbiology, so understanding the mechanobiology of bacteria. These are nanofluidic systems that we use to squeeze individual bacteria and see their responses. You can see some of the bacteria here trapped in our systems. And recently, we've been entering this new field called engineered living materials, where we use everything we've learned about how cells are inside bone matrix and maintain bone matrix, and try and recapitulate that in an engineering material you might use for a building. So we're really excited about these topics. But when I present this work and tell people about my history as a mechanical engineer and a biomedical engineer and somebody really focused on bone histomorphometry, I often get three questions. Why did you change your research direction to suddenly focus on microbiology? How did you choose the topic, in this case, microbiology? And then how did you do it? And so the talk is to go over my answers, what worked for me, or it seems to have worked for me. And hopefully you can all gain something from hearing my answers. And I have three quick answers to these questions. The first is, why did I change direction? Because I was imitating people who are smarter than me. The second is, how did I choose the topic? By staying off the beach. I'll tell you what that means in a minute. And how did you do it? I say is R&R, which is resources and resilience. So let's just dig in. First, I want to talk to you. There's lots of people who are smarter than me that I've been pleased to work with over the years. I want to tell you about two of them who really influenced my line of thinking before we started doing microbiology in the lab. And the first of these is Mitch Schaffler. I met Mitch at the ISBM meeting in 1999. That was my first ISBM meeting. And fascinated by the work he was doing. This is a section from one of his papers. At the time, he was studying how microscopic damage in bone could then stimulate bone remodeling. And so this is a very telling image from one of his studies showing a resorption space here, and you can see the microscopic cracks that are stained here stained here. And he and his team were able to show that that after the cracks were formed, resorption cavities came in and actually made a little osteon, even in a rat bone, which is not something you normally observe. And I was fortunate enough that he took me on as a postdoctoral fellow when I finished my PhD. And at the time, he was focused on studying the role of apoptosis in osteocytes as part of the signaling mechanism that would lead to this connection between microdamage and resorption cavities. And in hindsight, I didn't know this at the time, but if you think about it, in the 1990s is when apoptosis research really started booming. It had been recognized for some time, but there was lots of rapid growth, especially in association with cancer. And Mitch was the first one to show this, I believe the first one to show this in bone, the relevance of apoptosis mechanisms as part of the signaling process. And he showed it in osteocytes, he and his team. And I think what he did is he really captured this thing that was hot in the field, or had been hot for almost 10 years, and brought it into the bone field and made it something that most of us think about often when we think about bone. The second person I'll mention here is Linda Bonewald. I had the pleasure of meeting Dr. Bonewald at the 2001 Sun Valley meeting for the first time. Linda, of course, is a well-known bone biologist, but at the time she was starting to expand her research into osteocytes, and she gave what I consider a cornerstone talk on osteocytes at that meeting that has really influenced my thinking even now, or more than 20 years later. So this is one of the images from her lab. And if you watched what Linda went on to do in the 2000s, the field of autophagy, our understanding of how autophagy occurs in cells, was maturing and really accelerating. And Linda, I remember seeing at the ASVMR one of her talks, and by 2010, she was one of the first people to bring autophagy into bone research in a way that we all, I think, recognize today. And so we have two of these people, very smart people, very friendly people too, who saw what was coming, saw this exciting advancements in other fields, and brought it to the rest of us in the bone field over a space of 6 to 10 years. So, after I got all my research going in my own lab, we were, worked out all these visualization approaches. It took us 4 years took us 6 months to make the machine that would collect the images, and another 3 years to write the software that would allow us to make histomorphometry measurements, like the ones you're seeing here. But there was just one problem, and number one is that the method was extremely limited because it required too much labor, lots of very specialized technology, and it was slow. So to collect one of those images might have been 5 days of image acquisition. And if you start talking about one specimen as 5 days, that's just too long. There's only so many samples you can do in 1 year. And so that was one of the problems. The high labor cost and the technology costs associated with that advancement also prevented any other labs from being interested in taking up the technique. And so that was one of the limitations I realized down the line. The second was the studies we were doing. We do these great studies correlating, bone formation, osteocyte counts, and mechanical stress and strain. Our findings were, for the most part, telling us what we already thought were happening or confirming what we already knew. And as Mitch told me, well, what are we learning from this? I'm like, well, we're learning that what we estimated from a 2D slide was correct. And so at the time, I was like, well, we're going to have to do something different. We need to do something new because there's not much of a future for my lab if I keep confirming what I thought I knew beforehand. And so I spent a lot of time thinking about where we might go and what we might do. I had some people suggesting that we go into cell biomechanics. I heard a lot of people studying cell biomechanics making great discoveries in that space. And every time I thought about doing some cell biomechanics work, the thing that kept hitting me was the fact that there were so many other people who'd been doing the work for many years before me. I'd never done in vitro culture of mammalian cells in the lab. We never did any of that. And I just felt like I'm going to come in and try and compete with people who are more experienced and been doing a lot longer than I have. And so, but I kept hitting it in my mind. And then I went and saw a seminar at my university. I was at Case Western Reserve University, and we had a special visitor, Subra Suresh, who at the time was Dean of Engineering at MIT. He went on to be president of Carnegie Mellon University and president of, I believe, National University of Singapore, very renowned person. And he's really known for his work in material science and understanding how cracks move through materials like metals and ceramics and rocks. Literally, he wrote the book on it. So there's a copy of his book. It's still used as a textbook for graduate courses in material science and in mechanical engineering. But he came to give a talk, and he didn't talk at all about cracks in metals and ceramics. Instead, he spent his whole talk talking about the biomechanics of red blood cells. And this is an example of some of the work his group did looking at the mechanics of red blood cells. They used optical traps to grab onto a bead, and then would use that bead to tug on the red blood cell. And I vividly remember his talk. He also used microfluidic systems to squeeze the red blood cells and watch how they deformed as they went through microscale channels that sort of mimicked the vascular system. He was asking this question: how do red blood cells actually squeeze through these very small capillaries in the body? And after the talk, I remember I was just so excited about everything he'd said. He gave such a wonderful talk, but I couldn't get out of my head, this is the guy who became famous studying how cracks move through metals. How is it that this guy started doing red blood cell mechanics and studying red blood cells? And a little later, I realized that he picked the one cell in the human body that you don't have to culture. You just get it from the blood bank, expired maybe. And the one cell in the human body that really has no internal structures. Remember, there's no nucleus or complicated cytoskeleton the way we have in other cells that we normally think about for cell biomechanics. And so I thought to myself, well, if he was very clever in picking which model to work on. What else is there out there that's easy to culture and has no internal structure or no cytoskeleton, at least not a cytoskeleton like mammalian cells? And I said, that would be bacteria. And so that was my first inkling that we would start working in microbiology. So it sort of leads into, how did I choose the topic? And, I've been thinking about microbiology for some time, and after hearing that talk by Subra Suresh, but there's a high ramp up, right? I'm not a microbiologist. I'm a mechanical engineer. I had next to no microbiology background. How would I decide to do it? And the reason I did it, and the reason I say that, how do you choose a topic like this, is to stay off the beach. This is an analogy I got from Neil Pasricha. There's a copy of his book, You Are Awesome, up on the right-hand corner. My lab group read it last year for our retreat. And in this book, he reviews a lesson he learned in business school. He claims it's one of his most important lessons he learned. And he went to the dean in the business school and was asked him for some advice, like, what do you do? What do you do? And the dean described this situation. Let's say there's a fancy beach out there, and there's all kinds of beautiful people on that beach. There's several beautiful people on the beach, but you're not on the beach because you're waiting to be allowed in, because for some reason these beautiful people are allowed on there first. And you're waiting there, and you want to get in there because you want to go meet these beautiful people and spend time with them and get to know them. And the problem is that there's a long crowd of people next to you waiting to get onto that beach. And the advice that the business school professor gives him is stay off the beach, because if you go out there, you're going to have to compete with all kinds of other people. You're going to put all your energy into competing in this space. Why not instead find something else you can do that will have impact and be important? And so that's the lesson he took, Neil Pasricha took, and writes about in his book. And it's also part of the reason that we decided to go into microbiology. As I read more and more about the topic, I'd see articles like this. So see lots of these articles in Science and Nature about the microbiome, see them in mass media like The Economist. And it really got me thinking, like, well, there's a lot of stuff going on in microbiology and the microbiome, but who's doing it in the musculoskeletal system? And the second thing I noticed is that this is a plot that shows some of the timeline of microbiome research. So it goes all the way back to the first discovery of microbes down here in the 1600s. But what you're seeing here is this curve with the number of publications using sequencing approaches. And in 2005, next generation sequencing was introduced. And what that did was accelerate research in the microbiome. We'd known about the microbiome. We knew the microbiome is present ever since we figured out people started using antibiotics or discovered antibiotics. And they knew that if they gave antibiotics to farm animals, the animals would grow faster, mostly because of what the oral antibiotics were doing to the gut microbes. But by the 1980s, the field had slowed down because there was no way to measure most of those microbes. And it wasn't until next generation sequencing was introduced that the microbiome again became a hot topic, and the number of publications and the interest in the field zoomed, because suddenly we had a way of studying what microbes were there and what they were doing. So what is the microbiome? For those of you who aren't as familiar, these are the microscopic organisms that reside in the human body. We each have about 100 trillion of them on our body, on body cavities. The vast majority of the human microbiome is inside the gut. And each of us inside our gut houses about 1,000 distinct species that interact with each other and with our bodies, and that regulate our health. And we've seen effects of the gut microbiome in all kinds of different organ systems: Alzheimer's disease, cardiovascular disease, diabetes, and obesity. And so when I heard that, I thought, well, okay, so this microbiome thing influences all these other organ systems, and many of these organ systems are known to be correlated with bone and musculoskeletal disease. So there are probably pretty good odds that the microbiome is also influencing bone, either, if not directly, then through some kind of secondary mechanism. And when we started this, there had been, I believe, only one paper on the microbiome in bone. This is from Claes Olsen's group with Clara Sjögren who had done this first study looking at germ-free mice. These are mice that are raised in a bubble and have never been exposed to a live microbe in their entire lives, and compared to mice that had been germ-free but then got a normal gut microbiome. You can see that they've labeled this ConVR. And so they saw some clear differences, and this was another indicator to me that there was probably an effect of the microbiome in bone. So I had pretty good odds, even though I was working in this, I was not on the beach. I was thinking about working in this space that was not on the beach where everybody else was looking at cell biomechanics or other important factors. There was something off the beach, but I was probably likely to succeed. And so that's why we dove into the microbiome. And I had a sabbatical leave. I received a fellowship from the NIH called a senior fellowship from NIAMS. They support that mechanism, which is really just enough to pay for that to help support that sabbatical leave with my salary. I did a 3-week short course at Cold Spring Harbor in advanced bacterial genetics, and then spent a year in the microbiome lab led by Ruth Lay when she was at Cornell University between 2013 and 2014. And that really allowed us to kick off our studies of the gut microbiome and in bone. Now, I want to go over just a little bit of science here about what we found or how we did it. One of the things I learned during that year were every way we could possibly manipulate the gut microbiome in a mouse, because my group predominantly looked at animal models. And one way of doing this was to use oral antibiotics, putting antibiotics in the drinking water. And the microbiome community would often use a cocktail of 4 different antibiotics. You can see them listed here: ampicillin, neomycin, vancomycin, and metronidazole. And if you put that cocktail in the mouse's drinking water, it removes 99% of the microbes from the animal's gut. And when we did our first studies, we decided that probably wasn't a good idea to remove all of the microbes. So we just took 2 of these antibiotics, ampicillin and neomycin. And what we found is that changes the composition of the gut microbiome in the mouse, but the overall bacterial load is the same. And another nice thing about this is these antibiotics are not well absorbed at the gut lining. So neomycin has 0 oral bioavailability, so none of it gets into the bloodstream. And ampicillin has lower oral bioavailability. And what that did was allow us to do these experiments and be confident that it wasn't the antibiotic molecule itself that was getting into the bloodstream and influencing bone. It was probably something to do with the changes in the gut microbiome. And so what we did was our very first experiment. We took the mice at weaning at 4 weeks of age. We started them on the antibiotics and took them out to skeletal maturity at 16 weeks of age, and then looked at the bones. And Jason Gus, who's a graduate student leading this project, came back with the micro-CT data. So on the left is the trabecular bone volume fraction, and on the right is the femur length. And of course, he saw no differences, and he was so sad because he just spent upwards of 9 months to a year preparing this study and putting it all together. But he decided that he would do one last experiment and look at the mechanical properties of the bone before we would shut down the project and move on to something else. And we're lucky that he did, because when he did the mechanical testing of the bones in the mice, what he found was not a change in what he found was a change in the tissue strength, which is the material properties of the extracellular matrix. So again, the bones were about the same size and density, but the tissue strength was different. And so this was a really interesting finding from our perspective, again, as mechanical engineers, because it's the mechanical performance of the bone that we're normally worried about. And when we worry about regulating osteoclast and osteoblast activity, that's mostly so we can change the mechanical function of the bone, as opposed to our original interest in those cells. And so that has kicked off our lines of investigation. And now we're still working on understanding how these changes in the gut microbiome regulate the extracellular matrix in bone. And if you come to the ASBMR and ISBM, you can hear more about what we're doing in this space and our recent findings in this space and how we're trying to tease out that response. But to stay on to this topic of how we pulled off this switch, I want to go into this last question is, how did we do it? And again, I call it resources and resilience. And the first resource I will point out to you all is one that many of us many professors have, which is called tenure. And this is the situation that your position is guaranteed, your salary is guaranteed at the university, and and clearly, as long as you keep doing honest work, that you're going to stay at the institution. So I had a lot of job stability as a tenured professor. I will also mention that I, at the time, was a professor in an engineering school. And so my that's what we call in the US a hard money salary. So my 9-month salary was guaranteed as long as I did my teaching and service obligations at the institution. So I had a lot of personal stability financially when I decided to do this very different thing. The second thing I'll mention is my position at the time. I was at Cornell University in Ithaca, New York. Cornell is a spectacular place to work. It is also located in a small college town. And so I think you can see some of the pictures of the town in this photograph. There's the Cornell University Tower up here in the right. And because it's this small college town, distant away from the cities, the cost of living is relatively low. So one of the things I did, I'm willing to admit that I did, is I redirected any grant money I had for salary and moved it into the lab, because my teaching responsibilities covered my 9-month salary, and the cost of living in Ithaca was low enough that it was not such a big change in our lifestyles for me to take those what would have been months of summer salary support that would have come in as income for me, and redirect that toward the research projects that allowed me to move forward with this process. The next thing I'll mention are the spectacular students I had. I had a wonderful team of graduate students. At the time, my lab was really just graduate students. So mostly PhD students, some master students, and also large teams of undergraduates. The undergraduates during the term were not paid. They work for a course credit, so we pay them over the summer. But the other thing I was fortunate enough to have that allowed me to do this transition, or I would say empowered me to do this transition, were the number of external fellowships for my graduate students. So you can see out of this picture from the group in 2014, about half the students had an external fellowship from the National Science Foundation. And that really, that covers about 3 of their 5 years of graduate school were covered by this external fellowship. So if you think about it, my grant funds only had to support a certain number of the graduate students doing research in the laboratory, especially Marisol Luna, who was focused most of her work on the microbiome while she was there. So that's another thing that empowered my ability to make this transition. If you go back in hindsight, I want to point out to you, how the process happened. And first, I want to start with the grants. So my sabbatical ended in 2014. Immediately started putting in grants to the National Institute of Health, which is a major research funding agency in the U.S. And what I've done is stack these grants. These are a standard NIH grant. It's called an R01 for those of you who are not from the States. And then what I've listed here are some small grants. In our case, this is an R21 grant from the National Institutes of Health. These are much more modest grants that only last 2 years. And so you can see what I put in here are green are grants that were submitted and awarded, and the orange are grants that were submitted and not awarded. And then what happened was I would submit it and then submit it again and submit it again and submit it again and submit it again and submit it again. So what you're seeing is some of these little grants that are designed for high risk new ideas were coming through in this workshop, but each of them only had like 2 years of support. But the standard grant that was really expected for progress in the field, especially with something expensive like studying the microbiome where you have to send out the sequencing, that took much longer to get through. And you can see that it was finally funded in 2020 on the 6th submission of the same project. On the top, what I've shown you here is our publication rate, and the light blue are our review articles that we were publishing, and the purple are the experimental articles we were publishing. So you saw Jason Guss's first demonstration here of an effect of the gut microbiome in bone. And then these three stars are more studies of the gut microbiome in bone, as well as studies of the gut microbiome in osteoarthritis and infection of orthopedic surgery, in orthopedic surgery. So, timed it out. We had lots of preliminary data coming into lots of these submissions, but I think the grant reviewers like to see stuff that was published, and they like to see stuff that was established. The National Institutes of Health, for these large grants, these standard grants, they like to support work that has a good background to it. So they know the institution is going to get something, is very likely to get something from the financial support. So they really want to develop, they want lots of data developed. The argument here is that NIH grants, you typically want to have the first year of a 5-year grant done before you'd even consider submitting. And I clearly wasn't doing that here. In hindsight, I look back at this and I used to think, wow, it took so long to get these through. It took, if you think about it, that's a 6-year time period from starting to work on the topic until we actually had the funds to carry on the work. And you heard a little bit about how I financially supported the lab during that time. We still had bone mechanics stuff and histomorphometry stuff ongoing in the lab. That was another challenge, because I'm balancing our traditional bone biomechanics work with this new work in microbiology. My student was laughing at me one day because I think he said I would sit at the desk and sit at the desk in my rolling chair and talk about histomorphometry, and then I'd sort of roll my chair over to another desk and start talking about microbiome or thinking about the microbiome. So it was challenging, but I wondered, what's up with this time frame? Could I have done a better job? I kept thinking I probably could have done a better job. There's probably people who are smarter than me who could pull this off faster, and I know for a fact there are people who could pull something off like this off faster than I did. But then I looked at some other people who, again, who are smarter than me that I really respect. This is Fei-Fei Li. Fei-Fei Li is a professor of computer science at Stanford University, and she is also regarded as one of the world's experts in artificial intelligence. And Fei-Fei Li became super famous as a scientist and a technologist for her lab's development of something called ImageNet. And ImageNet was a collection of millions of images of everyday objects that were annotated. And it's that annotation. What was novel about it is they had such a large number, tens, maybe hundreds of millions of images that were put into this ImageNet, each annotated, and that allowed them to train machine learning algorithms to automatically detect objects by vision. And so when she did this, it ended up being transformative to the field of machine learning and artificial intelligence. And it's really the concept that's behind ChatGPT. Many of us use these large language models, is to have massive amounts of data that are then used. But when she started the project in 2006, nobody was doing massive amounts of data. Everybody thought they would discover the most clever algorithm, and that you don't need that much data. You just need to be more clever about your algorithm to make it. And she and her graduate students started making ImageNet in 2006. In 2009, they gave their first presentation of ImageNet, and nobody came to see the poster. Her graduate student had worked his tail off, annotating all these images, and nobody came to see it. You can read about it in her book. It wasn't until 2012 that people started to realize that what she and her team had created was actually a major discovery in how to do artificial intelligence and machine learning. We read this book with my lab this year for our retreat, and I asked them, "What do you think that graduate student was thinking between 2006 and 2009?" He's working his tail off, and everybody's yawning when he presents it at the conference. I think the laughs for him right now, because I'm sure he's doing very well. Another person I want to tell you about who I really respect is an old classmate of mine, Lindsay Marr, who Lindsay is an environmental engineer, and she did her PhD and her postdoc studying pollution caused by vehicles. So very interested in air pollution, especially the pollution caused by vehicles. And in 2003, she started her laboratory focusing on how air pollutants come out of vehicles and how they float around the air, where they can, of course, be detrimental to human health. But 10 years into her laboratory, she received an NIH New Innovator Award. This was before the NIH had early stage investigator. So even though she'd been out and running her lab for some time, she had never done NIH research before, and so qualified for a New Innovator Award. And this New Innovator Award was inspired. She's from California Central Valley. I'm also from California Central Valley. And there is a disease that's endemic to the Central Valley. It's called coccidiomycosis, commonly known as valley fever. And what it is it's a fungus that is native to the soil in those regions, in the American West and Southwest. And every so often, the spores of that fungus get kicked up into the air. And if you're unfortunate enough to inhale them, you might develop mycosis, which could be a short-term illness, fever, et cetera, but the cocci can also sneak into your body and become resident inside bones and joints. This can actually cause arthritic problems down the line. And so she thought, well, I'm going to take all this knowledge I have about how air pollutants come out of cars and apply this to diseases like cocci to try and solve some problems for people. Well, as a result of refocusing her laboratory on these airborne diseases, by 2020 she was the world's expert in aerosolized pathogens. And of course, the pandemic hit, and then her expertise was extremely needed worldwide. And so she was extremely influential in the nation's response and scientific response to COVID-19. There's a great article on the challenges she faced in the Atlantic called "We Need to Talk About Ventilation," because the medical literature had made a mistake. Somebody long ago had put a sentence in a medical textbook that specifies the size of a particle that could be aerosolized, as opposed to distributed in droplets. And she and the other engineers working in the space knew about it and had known about it for 20 years, but still nobody in the medical community was willing to accept what the engineers were saying, and she had to go through that fight to help fix that problem. And in recognition of this spectacular work she's done through her career, this last year she was named a MacArthur Genius Fellow and was also inducted to the National Academy of Engineering. So the message I want you to get from Feifei Li and Lindsay Marr is that in both of these cases, we're talking about a 6 to 10 year time period between the shift in research direction and major recognition of what happened. So when I looked at the time period it took my lab to move into this microbiome space, it ended up being very typical. So even these people who I think are much smarter than me, it also took them about the same amount of time. And I think that just says something about who we are as scientists and peer reviewers. Most of us, we're trying to do our own jobs, and we're also trying to review these other works, and it's hard to keep track of what everybody else is doing. In the business community, they say that if you switch your professional direction like this, it can often take people about 5 years to actually recognize you for that new thing you do. And I would argue that's what happened to us when we started doing microbiome work. It was about 5 years in, people started saying, oh, that Hernandez, that's the microbiome guy, as opposed to that's the guy who breaks bones and does very detailed measurements of bone histomorphometry. All right, so let's get back to our problem. We're thinking about osteoporosis and osteoarthritis and low back pain. We're thinking about musculoskeletal disease and what we can do about it. And I think there's a lot that the ISBM community can do. The number one thing, as I talk to people in other fields, that is holding back the musculoskeletal research community is the speed at which we get our results. Almost all of our results are image-based or histology-based. This is some work I'm doing with Kelsey Collins, looking at osteoarthritis in mouse joints. And then at the end of the day, you spend all this time sectioning the samples, and then somebody has to go through them and score the slides. There's all kinds of questions like, how many slides do you use? What's the correct orientation of the slide to take? The same questions the histomorphometry people were asking in the 1980s. And then at the end of the day, you get results like this, which are kind of scattered all over the place. And it's really hard to detect differences between groups, or you can detect big differences between groups maybe, but the subtle differences, which are probably more realistic to model the human condition, are really hard to see. What we need are better ways of measuring this, things that are faster, things that are more quantitative. And if you want an example from bone, anybody who's done the dynamic histomorphometry before has seen how long it takes to embed specimens undecalcified, and then the expertise required to perform those fancy histomorphometry measurements, which I believe there's a workshop at the ISBM meeting where you can learn how to do some of that. But it's a labor-intensive technique, and I would argue that bone really took off when micro CT came along, and then you no longer needed to all the embedding time, and you no longer needed the specific to be as skilled to make the measurements on a micro CT image as you do for the dynamic histomorphometry measures. I also work in orthopedics, and I just like to point out to people that probably the greatest innovation in orthopedics was the development of arthroplasty. This is Sir John Charnley, who was the surgeon who invented hip arthroplasty. He did a lot of experiments really on patients, figuring out what would be the best hip replacement design. And pretty much the designs that we use now are pretty much the same as the ones he came up with in the 1960s. Is that great? Yes. But I think we could do better. And I think we could have a lot of impact changing the technologies that we're doing now. This is a list of the top 10 inventions that shaped modern orthopedics. And I put the years that those inventions were introduced in parentheses to the right. And so you can see from this list, the most modern of these were the 1970s, the introduction of MRI. And don't get me wrong, there's been improvements in all of these techniques, but those improvements have been small incremental improvements that make things better. But I think we're at a point now where we could really stand for some real advancements. And I think the histology, the histomorphometry, and the visualization, quantitative visualization is what's going to allow that to happen. So if there's three things to take away from this lecture, think about imitating people that are smarter than you or more experienced than you. Stay off the beach, and think about the resources and resilience it will take to do a big shift, because it can be harrowing and it may not necessarily be for everybody. With that, if you want to hear a little bit more about innovation and how social status might influence innovation, take a look at my TEDx talk entitled Who Gets to Innovate? Oh, and come to the ASBM or ISBM meeting to learn more about bone, to learn how to make these technologies happen. And this is a picture of my group. Thank you.